Thank you for the introduction, and thank you to the organizers to let me be here. Well, to start, logical modeling or logical models are classified as qualitative models. So these allow us to integrate several information in order to study complex biological systems. Logical models are also known as Boolean networks. So this gives us insight into the dynamics of the respective system of our interest. Logical models are composed by nodes and arcs, and each node can be either a gene, protein, mRNA, or wherever you need it to be. And the interactions between them are given by the arcs, and this can be negative or positive. The interactions between the nodes are given by logical operators that could be not, and or. And once we have like the components of our model, we set an initial state, and as a Boolean model or logical model, the states are 0 or 1, 0 meaning inactive and 1 active. To construct a logical model, first, we go into the literature and search everything it's known about the system that you are interested, and then we construct a gene network to later perform simulations and model predictions. With this, we compare with experiments, and those in agreement, we can perform new simulations, and those in disagreement, there is room to add new regulations. And this information is possible to obtain from the epigenomics. And then we add it to the model and continue performing simulations. So tolerogenic dendritic cells are the ones that are responsible to polarize Tregs. And these Tregs are responsible to turn off the inflammatory response. So that's what is important in several autoimmune diseases. Tolerogenic dendritic cells can be obtained in the laboratory from the monocyte when we cultivate it with interleukin-10, interleukin-4, and GM-CSF. And also, it's possible to obtain dendritic cells that when we incubate it with the LPS, then they are going to be mature. So it's going to be like the counterpart with the tolerogenic dendritic cells that are going to turn off the inflammatory response. And there is one only differentiated between the dendritic cell normal and the tolerogenic dendritic cell, and this is the interleukin-10. So the tolerogenic dendritic cells has specific markers and a specific characteristic. They are characterized by exhibit low levels of pro-inflammatory cytokines and markers for dendritic cell maturation. They have high levels of anti-inflammatory mediators, also chemiosin and chemiosin receptors, metabolic control, and immunomodulatory molecules. So the aim of our work was just to build a logical model using healthy tolerogenic dendritic cell and identify key players in the tolerization cellular process. The methods that we use, we perform a macroarray analysis, then the identification of transcription factor binding sites, the logical model implementation using GeneSim, and the logical model testing using the notebook in Colomoto. For the microarray analysis, we look into the GEO database and we identify one data set that has samples for tolerogenic dendritic cells and modices. And the differentiation for the modices was done using interleukin-4 and GM-CSF, and for the tolerogenic dendritic cells was used interleukin-10, interleukin-4, and GM-CSF as we see in the first part. And there is an information or RNA-seq data from the tolerogenic dendritic cells with interleukin-10 protocol tolerization. So the results of our macroarray analysis were that we have several differential expressed gene in the tolerogenic dendritic cells as interleukin-10 and also CD14, that is a marker from the monocyte that it's the predecessor cell. And we have also some underexpressed genes in the tolerogenic dendritic cells that correspond to the maturation markers of the dendritic cell. So this is perfectly good. And we also perform a PCA plot in order to look cluster from the tolerogenic dendritic cells and the modices. We also identified 5 transcription factors that were overexpressed in the tolerogenic dendritic cells. And 3 of them, these first 3, TCF7L2, CP2L1 and GAL4 haven't been related with dendritic cell behavior at all, and there is also IRF8 and CBPB. So then we move to the identification of transcription factor binding sites using those 5 transcription factors overexpressed in the tolerogenic dendritic cells. And we took the matrices of all these transcription factors and look into the 2 kb upstream of the transcription start sites of the markers specific for the tolerogenic dendritic cells that we see earlier. We also look for a chip-seq done with the transcription factors of our interest into the remap database. And we found that was one chip for the TCF7L2 in the CD14 cell line. There is no chip for TFCP2L1. There is one chip for IRF8 into the THP1 cell line. CPPB has one chip into the monocyte, and GAL4 we didn't found any chip. And the results of the look of the transcription factors binding sites for the transcription factors we have in the y-axis and in the x-axis are the genes related to the tolerogenic dendritic cells. And the ones in color are the binding sites that we found. And with the stars are the sites that were corroborated with the remap data. Then we move to the logical model implementation into the gene sim. And in the first figure in the panel A, we have the data that we use to construct our model. First, there is the literature search, then the pattern matching that we done with the matrices from the transcription start sites, the gene expression analysis, and the signaling cascade that came from the interleukin 10. We put it together and we build our model that starts with interleukin 10, then the signaling cascade. In the yellow nodes are the transcription factors, the red nodes are the genes related with the inflammatory response, then some others for the signaling cascade, and the nodes in green are the corresponding for the tolerogenic behavior. Then we perform the logical model testing. So once we complete our model, we analyze and we wanted to know the differential or the final states that we can obtain with our model. And we have on the y-axis in this figure, all the nodes, and we have 2 columns. That one corresponds to the modices and the other corresponds to the tolerogenic dendritic cells. And we can see that the light blue belongs to the level 0 and the dark blue belongs to the level 1. So in the absence of the interleukin 10, we obtain the modice. So this is good. And also in the presence of interleukin 10, we obtain the tolerogenic dendritic cell behavior. And there is also what I show you in the figure on the right, where is the interleukin 10 that is present and then or the tolerogenic genes related. With this, we perform several mutations, and by mutations I mean that we force the value of the transcription factors into 0, and any condition can change that. And we can see that the nodes in black, they are active, and those in yellow are inactive. And we have 2 big clusters. The first cluster is for the inflammatory response, and the second cluster is for the tolerogenic behavior. And in the mutation of STAT3 with the presence of interleukin 10, we see that all the inflammatory genes are active and any of the tolerogenic dendritic cells genes are active. So this is affecting completely the tolerization. And also for TFCP2L1, there is also some inflammatory genes, and we lost some of the tolerogenic genes. For TFC7L2, in the presence of interleukin 10, we also lose some tolerogenic genes. And for GAL4, we have the same phenotype, that we lose some tolerogenic genes. With this, we conclude that STAT3, TCF7L2, and TFCP2L1 and GAL4 are key players for the tolerogenic dendritic cell commitment. Then Then we wanted to know which percentage of the population was committed to the tolerogenic dendritic cell behavior. So to do this, we perform a simulation through the time, where in this graph I show you the percentage of the population that it's committed to the heterogenic dendritic cell behavior in the presence of interleukin 10. And in the y-axis is the percentage, and in the x-axis is the time. And we can see that have some curves that grow and then decay, and then that we have only one curve that reaches the 100% of the population and that exhibit the specific markers for the tolerogenic dendritic cell behavior. And as conclusions, our model gathers specific markers for tolerogenic dendritic cell gene expression and predicted transcription factor binding sites. We test our model on the different mutations and predicted the role of STAT3, TCF7L2, GAL4, TFCP2L1 to the tolerogenic dendritic cell commitment under interleukin-10 differentiation. And our model can be a base to perform experimental corroboration and integration in the different tolerization protocols. And I would like to thank all my lab mates, the technical support that I receive, and also the institution that allows to perform this analysis. And I'll give you my email and my Twitter account so we can be in touch. And I'm ready to take your questions.
Thank you. Thank you, Karen. Nice talk. Any question for Karen? I have one. How do you, sometimes, monocytes are also related to macrophages.
Yeah.
In your cell protocol to obtain these dendritic cells, do you see some macrophage feature, or is always, it's clear that it's a dendritic cell?
Yeah, it's clear that there are dendritic cells because of the use of interleukin-4. It has been reported that interleukin-4 allows the activation of STAT6 in order to obtain the dendritic cells, and STAT6 is blocking the expression of macrophage-specific genes.
Okay.
Yeah.
Thank you, Karen.